Welcome back to Exercise Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to the channel for future videos and notifications. We're going to do two things in this video. One, we're going to very briefly talk about glucose entry into a skeletal muscle cell and how it gets metabolized, not in any detail really about the actual pathways, but just show generally what's going on. And then we're going to compare that to the same thing, but for fatty acid metabolism. So we're mostly going to focus on the fatty acid component because we've covered glucose in other videos. That's the first thing. The second thing is really just kind of an overarching concept that hopefully you'll be able to see as we go through this, that it's a lot more complicated for fatty acids to get into the cell and ultimately into the mitochondrial matrix. And that kind of leads to some implications with the efficiency of metabolism. What we're ultimately going to see is that the efficiency of glucose metabolism for energy is very high efficiency for glucose because it's fairly easy for any cell to do it. Fatty acid metabolism still occurs, and I mean, it's not like the cell can't do it, but in terms of just overall efficiency and time that it requires, mainly time, it's not the preferred source of energy. Even though it's going to occur pretty easily, I mean, relatively speaking, it takes more time. And so that's going to play some implications in a future video where we're actually going to talk about how fuel source varies with exercise intensity and duration. So make sure to look out for those videos. But for now, we're just going to go over the general pathways of what's going on. But first, let's begin with glucose. So let me orient you with the picture. Here we have the plasma membrane. Out here's the extracellular space. And then here is the cytoplasm or sarcoplasm, okay, which is the cytoplasm of the muscle cell. This whole thing right here, which I could really color these, the typical color of the mitochondria, and that's yellow. This all in here is the mitochondria. This is the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. And the space between them is the intermembrane space. And the central part of this, all in here, where all this business is occurring, is the mitochondrial matrix, or just called the matrix. All right, so glucose is going to come from the blood, at least the plasma glucose that is, and it's going to enter into the cytoplasm through a glucose transporter, usually abbreviated GLUT. Okay, once glucose is in the sarcoplasm, it's going to, for the most part, immediately go into glycolysis, and we know that the end result of glycolysis is pyruvate. Okay, it is true the glucose can also enter the pentose phosphate pathway, but that will also immediately consume it. But the major pathway is glycolysis to produce pyruvate. Pyruvate, indirectly, it's going to undergo a couple reactions that are not really important here, but ultimately pyruvate ends up inside the mitochondrial matrix, where this enzyme PDH, also called the link reaction, pyruvate dehydrogenase, converts that pyruvate to the central coenzyme acetyl-CoA. Now, what I want you to notice is, just with that already, we're already at acetyl-CoA. We're already in the matrix, we've already hit acetyl-CoA. That acetyl-CoA then is just going to go into the TCA cycle. Okay, notice it didn't take me very long to explain all that. Now what I want to do is focus on the fat metabolism, okay, and how fat gets into the cell, and then ultimately how it gets into the matrix, and you're going to notice it's a lot more complicated, okay? So usually we're going to have a fatty acid transporter. Good examples of that are going to be the FAT, also called CD36. That just stands for fatty acid transporter. There's another type which I haven't mentioned here that'll also work, and I should probably put it just so you have it. That's actually called fatty acid binding protein, FABP. It's going to be a similar membrane protein that's going to pick up fatty acids and move them into the sarcoplasm. That's another one that'll do it. If you have a medium chain fatty acid, they don't even need a transporter. They'll just diffuse across the membrane by simple diffusion. But by some mechanism, the fatty acid has to end up inside the sarcoplasm. Now, once in the sarcoplasm, there's an enzyme here called fatty acyl-CoA synthetase. If you've taken a biochemistry course, you've probably mentioned this enzyme. This enzyme is going to conjugate the fatty acid to a coenzyme A. So we get a fatty acyl-CoA. Now, this is where the complicated stuff begins. The fatty acyl-CoA has to get into first the intermembrane space and then the matrix. But it's a complicated thing. So the fatty acyl-CoA is going to move into the intermembrane space, but not with the CoA. it's going to hit this enzyme, which is in the outer membrane of the mitochondria, called CAT1 or CPT1. Now, what CAT stands for is carnitine acyl transferase. If you use the CPT nomenclature, that stands for carnitine palmitoyl transferase. The reason that sometimes they'll use CPT for carnitine palmitoyl transferase is palmitoyl is for palmitate or palmitic acid, which is the most common fatty acid. 16 carbons is the most common fatty acid, so you just call it that way. But this enzyme will do this with any length fatty acid. And so carnitine acyl transferase 1 is going to take this fatty acyl-CoA from the sarcoplasm, and it's going to convert it into a fatty acyl carnitine. In other words, what's happening is the coenzyme A group that was on the fatty acid in the sarcoplasm is going to be substituted out for carnitine. And I highly recommend you go watch the video that I actually have explaining all the details of carnitine, how it can be made de novo through cellular biosynthesis, and ultimately what it's going to do I explain in this video. But here we're just going to take my word that the CoA gets substituted for this carnitine on the fatty acid. Okay, and so we have a fatty acid carnitine joining right here. That's going to be able to move into the intermembrane space. And what's going to happen is this fatty acyl carnitine is then going to be transported into the matrix. Now, I don't have it shown here explicitly, but what's going to happen here, the fatty acyl carnitine is going to be transported by this translocase, that's this T right here, into the matrix, and then immediately it's going to react with CAT2 or CPT2, which is carnitine acyl transferase 2. I'll go ahead and brief you on this right here. This is the molecule carnitine. This is actually its molecular structure. So when the fatty acid needs to be moved ultimately into the intermembrane space, the CAT1 or CPT1 is going to transfer the fatty acyl group, which is this over here, onto this hydroxyl of carnitine. Okay, so this is the carnitine with the acyl group attached. This is how it's going to move in the intermembrane space. Right here, fatty acyl carnitine. But as soon as it's transported into the matrix through the translocase, the reverse reaction is going to occur by CAT2 or CPT2. Okay, that fatty acyl group is going to be switched back to a coenzyme A, regenerating carnitine. Okay, so notice here, once the fatty acyl carnitine is translocated into the matrix, immediately that carnitine group is going to be swapped out again for the CoA, and we're once again at a fatty acyl CoA, which is essentially what we had in the sarcoplasm. Okay, but in order to transport this into the matrix, it has to go through this intermediate step where the CoA is substituted for a carnitine, and then it's substituted back for a CoA. Seems like a complicated process that could be avoided, but that's just the way it is. Okay, so at this point, now we have a fatty acyl CoA in the matrix of the mitochondria. So now that the fatty acyl CoA is in the mitochondrial matrix, beta oxidation can occur, and we're going to get three things from this, not necessarily in this order, but we get NADH out of it. One of the enzymes of beta oxidation gives us NADH, which is going to power complex 1 of the mitochondria. We also get, and it's actually the first enzyme that does this, we actually get an FADH2. Now, I have it here like it looks like it's going to power complex 2. That's not actually true. It doesn't power complex 2, but there's another enzyme in the respiratory chain that's not usually included in this list of 4 that's very similar to 2, and it's actually going to power that one. I actually have another video in the Electron transport chain playlist where I discuss that process. But in any case, FADH2 is going to power the electron transport chain. We also get acetyl-CoA, which is the central coenzyme. Now, glycolysis, very easy. Get the pyruvate in here and convert it to acetyl-CoA. In the case of the fatty acid, we have to go through conjugating it to CoA, moving it across this membrane, and substituting that for carnitine. Then we have to move it again into the matrix, substitute it back for coenzyme A, undergo beta oxidation just to get that acetyl-CoA. So for fat, it's a lot more complicated. Even though there's more enzymes involved in glycolysis than all of these, Glycolysis is very easy to go through. This process will actually take longer for fat. But in any case, the acetyl-CoA that we produce is then going to go into the TCA cycle. And I didn't mention this before, but regardless of the source of acetyl-CoA, we're then going to get NADH and FADH2, which will then power the electron transport chain. This complex over here, which I have designated Roman numeral 5, this is just kind of an older nomenclature, but just for the sake of space in here, I couldn't write it. This is ATP synthase. And remember, the ultimate purpose of the electron transport chain are these proton pumps, which are powered usually by NADH or electrons that are indirectly from it. And protons get pumped into the inner membrane space, and those protons create a gradient which powers the phosphorylation of ADP to make ATP, which is our energy source. Okay, so for both glucose and fatty acids, they undergo completely different modes of metabolism. Okay, for the most part, glucose's metabolism is in the cytosol, in the sarcoplasm. It's glycolysis. For fatty acids, the majority of the metabolism's in the matrix, beta oxidation. Ultimately, we get the same thing. We get acetyl-CoA, which goes into the TCA cycle or Krebs cycle, as some people will call it. But overall, that's for the synthesis of ATP for the muscle cell. Now, the one thing you should understand is that the fatty acid metabolism overall is not as efficient. Okay, that's partially because of this process. It's also because a lot of the fatty acids are actually going to have to be mobilized from adipose tissue. And that process also is going to take a little bit longer. So the key here is that if you're a muscle cell, glucose is going to be a nutrient that's a lot more efficient to metabolize. Fatty acids are going to be less efficient. Now, I'm not going to go over this in much detail now. We'll see this in a future video. But as exercise intensity goes up, as we start exercising harder and harder, our muscle cells, they're going to need to be generating energy much faster, more energy and faster. They're going to have to rely on a more efficient energy source, okay? So at low intensities, fatty acids are going to be predominantly where they get their energy, because at low intensities or rest, you don't need the energy as quick. You don't need the ATP as quick. You can suffice with a less efficient energy source. As we start going up to 80%, 90% of our VO2 max, very intense exercise for muscles, they're going to need that energy quickly and a lot of it. So it's going to be more... it makes more sense to rely on a more efficient mode of metabolism. And so at high intensities, we're going to rely much more percentage-wise on glucose. Okay, we'll cover that in a future video, but hopefully this process, the physiological processes and the biochemical processes depicted here, make sense. These are just the basics. If you want more detail on these processes, such as glycolysis, the linking reaction and beta oxidation, or the electron transport chain, I have plenty of playlists on those on this channel. Please like this video and subscribe to the channel for future videos and notifications. Thank you very much.